So good day to everyone. We are here to discuss about the topic lac operon, which is a critical example of gene regulation in case of prokaryotes. So let us discuss about the lac operon. The lac here means the lactose, okay? And lactose is composed of glucose and galactose, okay? So we know that lactose is a disaccharide. Which is made up of glucose and galactose by beta 1,4 linkage. Understand? Now, and operon means it is a cluster of gene. What? It is a cluster of genes which works under a common promoter. Got it? So, operon is a cluster or group of genes which works under a common promoter. Understand that? Now, let us draw a diagram or representation of lac operon gene. So, lac operon consists of 3 parts. How many parts? 3 parts. Okay. First one is regulator. Second is promoter. And third part is structural gene. Got it? So, let us discuss them one by one. Regulator gene consists a DNA sequence of lac I gene. Okay, lac I gene forms a compound known as repressor. Repressor is a homopolymer. Homopolymer, that is homo means same subunits. How many subunits? 4 subunits. Homopolymer of 4 subunits. Then promoter contains a sequence in it known as operator. And promoter is the region where RNA polymerase attaches to regulate, or we can say that attaches to start transcription. We all know that transcription. What do you mean by transcription? Transcription is the formation of RNA from DNA. Okay. Now, structural gene here is composed of 3 structural genes, that is lac Z, lac Y, and lac A. Okay, so these on transcription form a 5' to 3' mRNA, and we say that it is a polycistronic mRNA. Poly means many, cistrons means many cistron, and cistron is the smallest functional unit, which can further form a functional protein, functional product. So here lac Z forms beta galactosidase enzyme. Lac Y form beta galactosidase permease, beta galactoside permease, and lac A form trans acetylase. So this is the structural part of the lac operon. Now what we say, if a prokaryotic cell is grown in a medium, this is a prokaryotic cell, let us say this is a prokaryotic cell and grown in a medium of glucose, then what happened? No function of lac operon. That means there is non-functional operon. Lac operon is non-functional in the presence of any medium other than lactose. We can say that lac operon is non-functional if the medium, if the substrate present outside the prokaryotic cell is other than lactose. Simply we can say that, okay? And if we grow a prokaryotic cell in a medium of lactose, then only the lac operon is functional. Okay, lac operon is functional only in the presence of lactose, that is it. And thus lactose act as inducer. We can say that. How? Because lactose is that product which causes the induction, which induces the function of this lac operon. That is, lactose is the only substrate which induces the function of these structural genes, these lac operon, under RNA polymerase. That is it. So we can say that lac operon is an inducible operon because lactose presence causes the functioning, or we can say that induces the functioning of lactose operon, that is lac operon. Okay, now we further study that how these genes functions, or how lactose operated the function of these genes, this operon. So now we start with, if lactose is absent, where, that is, if we grow a prokaryotic cell with no lactose in the medium, that is, if we grow a prokaryotic cell in a medium with no lactose, then what happened with the lac operon? Is it function or non-function? Let us say that. Then RNA polymerase comes to to transcribe this DNA molecule. Got it? RNA polymerase. It transcribes lac I mRNA, and this mRNA forms repressor because there is no hindrance for lac polymerase, RNA polymerase. It goes in this direction, and while going from this direction to this direction, it transcribes this regulator gene, that is lac I gene, and it forms the lac I mRNA, and mRNA in after translation forms a repressor. And we earlier said that repressor is a homopolymer, that is it is a tetrahomopolymer. And first case is, in the absence of lactose, what happens? Repressor is formed, and repressor in the absence of lactose gets attached with the promoter. Let us say promoter repressor attach in a way like this. Okay, when the polymerase reaches this site, there is a hindrance of repressor present at the promoter site attached with the operator. That is, repressor is attached on the operator site of promoter, and it blocks the pathway, it blocks the passage for RNA polymerase. So there is no transcription. Understand? So repressor here act as negative regulator. Understand? Repressor here act as negative regulator. How? Repressor act as negative regulator of lac operon, because in the presence of repressor, it attached with the operator of promoter, or we can say that it attached with the promoter and blocks the pathway of RNA polymerase. It does not allow the polymerase to move in this direction so that it can transcribe the structural gene. So we can say that repressor blocks the transcription of structural gene. Thus we call repressor as a negative regulator of lac operon. Got it? Okay. Now we study the second case. So now the second case is in the presence of lactose. So when we grow a prokaryotic cell in a medium with lactose, so what happens then? Lactose RNA polymerase attached with the DNA goes in a direction, transcribes the lac I gene, which form lac I mRNA, and it further form repressor. Okay. Now we say that repressor is a tetrapolymer, homopolymer. Now let us represent lactose by star. Now, lactose in its presence attached with the repressor and blocks inactivate. That is, you can say that lactose attached with the repressor and inactivate it. We earlier said that repressor's function is to attach the operator. A repressor function is to attach the operator of promoter, or it attached with the promoter and blocks the functioning of RNA polymerase. But now, lactose inhibit the functioning of repressor. Thus, lactose act as inducer here. How? Let us understand this. Now, operator or promoter remains free. There is no hindrance, there is no blockage of operator by repressor in the presence of lactose, and RNA polymerase keeps going on transcribing this segment, and it transcribes the Z gene to form beta galactosidase. Sidase means it is that gene which catalyzes, which breaks a substrate. And now here, what is our substrate? Our substrate is lactose. Beta galactosidase break down the lactose into glucose and galactose. Glucose and galactose, which is further used in metabolism. This is not our part of study today, but we can say that lactose is broken down by beta galactosidase into glucose and galactose. That's it. Okay. And when the RNA polymerase reaches the Y, it causes the formation of permease. Permease is the gene which allows, permease is the enzyme which allows the entry of galactosides. Galactoside, what is galactoside here? Here is lactose into the cell, and A gene transacetylase transcribes to form mRNA, which then upon translation forms an enzyme transacetylase. Transacetylase actually transfer the acetyl coenzyme A on galactosides. It is actually not a function of metabolism, it is a function of detoxification. So, the structure genes having three components, three small structure genes Z, Y, A. Z form the galactosidase, and galactosidase break down the lactose, that is substrate, into glucose and galactose. Permease allows the entry of lactose from the medium into the cell, into the prokaryotic cell, and transacetylase transfers the acetyl coenzyme A into galactosidase. Here, lactose, why we call lactose as the inducer? Because it induces, its presence induces the functioning, induces the RNA polymerase to transcribe the structure gene to their respective products. So, lac operon is an inducible operon. How? Lac operon is an inducible operon. So that's a short description about the lactose operon. Thank you very much.